All right, welcome to the first lecture of electron transport and oxidative phosphorylation. So, might as well just write that down here. And for future, I'll probably just write these as electron transport abbreviated and oxidative phosphorylation as oxphos. So, where ATP made in glycolysis and the TCA cycle is the result of a substrate level phosphorylation, NADH-dependent ATP synthesis is the result of oxidative phosphorylation. So what's the difference? Electrons stored in the form of reduced coenzymes, NADH or FADH2, are passed through an elaborate chain of highly organized proteins and coenzymes called electron transport chain, where they finally reach molecular oxygen, which is your terminal electron receptor. Ultimately, where we're going with this is that NADH or FADH2, which are reducing coenzymes, enter into electron transport where they're passed from protein to protein. And through this sequence of transfers, energy is harvested from these high energy electrons and subsequently are harvested and utilized to generate ATP and are ultimately transferred to oxygen, which is our terminal acceptor. So each component of this chain can exist in at least, as a minimum, of two oxidation states, and each component is successively reduced and reoxidized as electrons move through the chain, which we just described, and high energy electrons to our lowest energy state, where we've harvested as much as we possibly can. So in the course of electron transport, a proton gradient is established across the inner mitochondrial membrane, and it's this proton gradient which provides the motive force which allows for ATP synthesis. So, big picture, we're going to be passing electrons from our reducing equivalents through the electron transport chain to oxygen. This is going to establish a proton gradient. In the inner mitochondrial membrane. And what that looks like is, whenever you have a membrane or division and you have a gradient where you have a high concentration on one side and a lower concentration on the other, there's a natural tendency for the side with the high concentration to want to move across the membrane to the lower side. And if you provide a mechanism by which to do that, such as a pour or a pump, this energy can be harvested to produce a chemical in a fashion which does not happen spontaneously, such as the synthesis of ATP. So, big picture, C is use the gradient to make ATP. So ultimately what we're doing is taking these reducing equivalents and we are making ATP. So the process of electron transport and oxidative phosphorylation are inherently membrane associated. In prokaryotes, which are our simplest form of life, they typically consist of a single cellular compartment surrounded by a plasma membrane and a more rigid cell wall than eukaryotes. In such a system, the conversion of energy from NADH or FADH2 to the energy of ATP, the electron transport and oxidative phosphorylation, is carried out and across a plasma membrane. Now, in eukaryotic cells, which is what we're going to be focusing on, electron transport and oxidative phosphorylation are localized in mitochondria. So you know what? Let me make a little bit of space here. There we go. And this makes sense, because we're all familiar at this point that mitochondria are considered the power plants of cells, so it would make sense that the place where ATP is synthesized is localized to the membranes of mitochondria. And additionally, mitochondria are also the sites of TCA cycle activity and, as we'll see in future chapters, fatty acid oxidation. So all of these mechanisms, such as TCA, fatty acid oxidation, and electron transport and oxidative phosphorylation, are going to be mitochondrial processes that are all membrane. Well, not all of them are membrane associated, because some of these are not, but in terms of electron transport and oxidative phosphorylation, they're membrane associated. Now, mammalian cells contain about 800 to 2,500 mitochondria typically. Other types of cells may have as few as 1 or 2 or as many as 500,000 mitochondria. A good example, human erythrocytes, whose purpose is simply to transport oxygen to tissues, contain no mitochondria at all. So there's a high variability of mitochondrial density depending on the cell type or tissue type. Mitochondria, I guess we should spend a little bit of energy on this, are surrounded by a simple outer membrane and a more complex inner membrane. So if I were to doodle a mitochondria, we have an outer membrane, and then we have a membrane internally which has a lot of folds to it. And this is a gross oversimplification, but you know, it's my artistic skills. And here we have our inner membrane, and between them we have an intermembranous space and within the inner membrane we have the matrix. So when we're discussing the localization of different biochemical events, we need to be able to reference the outer membrane, inner membrane, intermembranous space, and matrix of the mitochondria. Now, the mitochondrial matrix, which we have here in the middle, inside the inner membrane, contains enzymes for the TCA cycle specifically, which is why I had to amend my previous statement. The space inside the inner mitochondrial membrane, or the matrix, and it contains most of the enzymes for the TCA cycle and for fatty acid oxidation. The exception being succinate dehydrogenase of the TCA cycle, which is located in the inner membrane itself, and it's integrated into electron transport. In addition, mitochondria contain circular DNA molecules along with ribosomes and the enzymes required to synthesize proteins coded within the the mitochondrial genome. Although some of the mitochondrial proteins are made this way, most are encoded by nuclear DNA and synthesized by cytosolic ribosomes. They just have their own genetic material, but that's a different topic for a different chapter to elaborate on. So, as we've seen, metabolic energy from oxidation of food materials, which include carbohydrates, fats, and amino acids, and proteins, all this metabolic energy is funneled into the formation of reduced coenzymes. And we've seen this funneling process in our earlier chapters. So we're going to say, typically, foods through multiple funneling steps go to reduced coenzymes such as NADH, primarily, and reduced flavoproteins, which are FADH2. The electron transport chain reoxidizes the coenzymes and channels the free energy obtained from these reactions into the creation of a proton gradient. So we need to take these, enter electron transport, oxphos, and get our ATP. Now, the reoxidation process involves the removal of both protons and electrons from the coenzymes. Electrons move from NADH and FADH2 to molecular oxygen ultimately, which is the terminal acceptor of electrons in the chain. And the reoxidation of NADH specifically looks like this, where NADH, which is our reductant, plus protons plus molecular oxygen, which is our oxidant, goes to form NAD plus H2O. And this involves two half reactions. So this is our overall. Let me scroll up a little bit here. And our two half reactions look like this. NAD plus 2 protons plus 2 electrons yields NADH plus a proton. And this has a standard, what's going on here? This has a standard reducing potential of negative 0.32 volts as drawn in the direction it's drawn. The other half reaction is, in its most reduced state, half an equivalent of molecular oxygen plus 2 protons plus 2 electrons yields water. And this has a standard reduction potential of positive 0.816 volts. So here, the half reaction shown second is the electron acceptor, and the half reaction first is the electron donor. So this is our donor, this is our acceptor reaction. And because of this, we can sum our two reduction potentials to describe the overall reduction potential of our overall reaction, which we described at the beginning, to mean that the change in reduction potential for the overall reaction is going to be 0.816 minus a negative 0.32 volts, and we're going to have a net 1.136 volts for this reaction. So, in addition to this, we can use the relationship between Gibbs free energy and reducing potential to find that because of this, standard Gibbs free energy is negative 219 kilojoules per mole. So molecules along the electron transport chain have reduction potentials between the values specifically of NAD plus and NADH couple and the oxygen H2O couple. So electrons move down the energy scale progressively from more positive reduction potentials. And this is kind of like a stepwise thing. When we look at talking about energy, whether it's Gibbs free energy, reduction potential, we're talking generically about a high energy system going to a lower energy system, less stable to more stable. If we think about energy on this axis and reaction progress on the y-axis, we're used to seeing diagrams like this. I keep going back and forth to them. However, here we're starting with high energy electrons, and we want to take them to lower energy. And as they're passed across from protein to protein, the energy of the electrons steps down stepwise as we go from our highest position to our lowest position, where we end up with water in our most stable, lowest energy conformation for those electrons. And along each way, we have deltas associated with these in which energy is harvested in a very, very efficient manner. The electron transport chain can be isolated into 4 different complexes. The first are our flavoproteins. And the flavoproteins contain tightly bound FMN or FAD as prosthetic groups and may participate in 1 or 2 electron events. The second one is coenzyme Q, or ubiquinone. I typically call, I just dragged that around. Let me erase that. Redraw it. Coenzyme Q. I always call it ubiquinone, or routinely call it ubiquinone. And this can also be abbreviated as coenzyme Q or UQ for ubiquinone. And this can function in either 1 or 2 electron transfers. There's several cytochromes. And these are proteins which contain heme prosthetic groups. And this includes cytochromes B, C, A, etc., and variations within that. There's a C and a C1, an A and an A3. And cytochromes are 1 electron transfer agents in which the heme iron is converted from Fe2 to Fe3 and back and forth. So this involves iron 2 and iron 3 being the mechanism by which single electron transfers take place. There are a number of iron-sulfur proteins. Which participate in 1 electron transfers as well. Also through the iron, going from iron 2 to iron 3 and back. And there's also protein-bound copper, which involves single electron transfer. And this takes place through going back and forth between copper 1 and copper 2. So all these intermediates, except for cytochrome C, are membrane associated, either in the mitochondrial intermembrane of eukaryotes or in the plasma membrane of prokaryotes. 3 types of proteins involved in this chain, including flavoproteins, the cytochromes, and the iron-sulfur proteins, possess electron-transferring prosthetic groups. Notice that the coenzyme Q, or ubiquinone, does not have a prosthetic group. It can accomplish this feat directly. Now, what does this look like in terms of what we talked about in the previous, I'm not going to call it a slide, but the previous part of our discussion? We talked about how electrons are passed from NADH, from complex to complex to complex within the electron transport chain, and how energy is harvested along each path. And we also talked about how the reduction potential of each of these proteins and prosthetics lays somewhere between our highest energy, NADH, and our lowest energy, water. What that looks like, crudely, let me make a little diagram. On this axis, we have our reduction potential in millivolts, with neutral being 0, 200, 400, 600, so about plus 200 plus 400 millivolts plus 600 millivolts, and then we have 200, 400 millivolts of potential. And complex 1, let's see here if I can do this justice. Need to bring this out a little bit. Has a sequence of transfers within complex 1 which range between somewhere around 600 and going all the way down to close to 0. Oh, wow. All the way close to 0. And the initial interactions take place around 400, and then it moves from NAD plus NADH to FMN, which will be here, and we're going to get into more detail here, and then we'll also see some transfers to iron-sulfur groups around here and here, and then we'll have another iron-sulfur group which has a transfer close to 0. Now, there will be a handoff between complex 1 and complex 2, and there's going to be an FAD, which has a just slightly negative potential. There's going to be the fumarate succinate dehydrogenase component, which lays about here. There's going to be some more iron-sulfur prosthetics here and here. And this is all ballpark. And then there's ubiquinone. Ubiquinone lies around here. So this is our complex 1, this is our complex 2. We have ubiquinone, which is going to be transferring electrons between 1 and 2 to complex 3. Complex 3 is going to lay somewhere bridging neutral, going all the way down to about minus 400. This is not beautiful, but you get the sense of it. Notice that there's going to be more steps where we have some transfers of electrons bridging neutral potential, and then several at plus 200 and change as well. And then we're going to have our transfer to complex 4, which is going to be around plus 200-ish. And we're going to have a copper interaction around here, and we're going to have our cytochrome A3 all the way down here. So the important thing is, here's our 3, here's our 4. Don't stress yet, we haven't gotten into the details of the complexes, but the big picture here, and we're going to go into nice amount of detail for complexes 1, 2, ubiquinone, 3, 4. The important point is that you know that when NADH comes in, it has the electrons at this negative voltage, this high potential, this high energy. There's a lot of electronic energy that we can harvest for the purpose of making ATP, but to sequentially harvest it requires the successive transfer of the electrons. To lower and lower units. And within each of those steps, we get that stepwise reduction, which I showed previously in their potential. And we can sequentially, deliberately harvest increments of energy from them until we get to the terminal state of electrons being housed in water, which is the most stable place for them to be in the system and the terminus for our sequence of reactions. So the components of electron transport chain can be purified from the mitochondrial inner membrane. Solubilization of the membranes containing the electron transport chain results in the isolation of 4 distinct protein complexes, which is why we keep referring to 4 different complexes, and the complete chain can therefore be considered to be composed of 4 parts. 1, NADH coenzyme Q reductase. Wow, I just keep finding new ways to utilize this program. 2, succinate coenzyme Q reductase. I have not had this issue before, but I seem to find new issues with technology every time I utilize it. 3, coenzyme Q cytochrome C reductase. And finally, 4, cytochrome C oxidase. There should be an A in there. There we go. So, complex 1 accepts electrons from NADH, serving as a link between glycolysis, the TCA cycle, fatty acid oxidation, and the electron transport chain. Let me make a little bit of room here. So here we have complex 1, and what it is, and this is our NADH coenzyme Q reductase. It's our bridge to TCA, glycolysis, and fatty acid oxidation, and it's going to be accepting NADH. I need to make this a little bit bigger because I need room to write inside of this box. It has some FMN and some iron sulfur centers. And what it ultimately is going to do is pass electrons from our NADH to our pool of ubiquinone. And we'll get into the pool of ubiquinone and why it's a mobile pool in just a second. And then our complex 2, let me make a little bit more space. Well, before we get to complex 2, let's elaborate a little bit. So both complexes 1 and 2 produce a common product, which is our reduced coenzyme Q, and our reduced one is our UQH2 because we have 2 protons associated with it. And this is the substrate for the coenzyme Q cytochrome C reductase, complex 3. Now, complex 2, which also feeds into ubiquinone, is going to have our succinate dehydrogenase, which is going to be our FAD equivalent. So complex 1, taking in NADH, succinate dehydrogenase complex 2, taking our FADH2. And this is also going to have some iron centers and some FAD binding units. But ultimately, we're going to take the energy from these high energy protons and be moving them towards ubiquinone. And then once everything's in ubiquinone in its reduced form, those electrons are going to be moved to complex 3. This is going to be our cytochrome complex. Which is then going to move those electrons to a cytochrome C, which is a mobile electron carrier, which will then transfer it to complex 4, in which ultimately a half equivalent of oxygen will be converted to water. Scroll this in so you can see the whole picture. And now what we're going to do is dive a little bit more deeply into the specific functions of complexes 1, 2, ubiquinone, 3, and 4. So complex 1, also known as NADH dehydrogenase, is going to take electrons from our NADH and move them to ubiquinone. Let me redraw that. Wow. I haven't the foggiest idea how I just did that. Let me scroll up and get that out of the way, because I cannot get rid of that every time I do it. I just did it again. And at this point, with all of the jargon and all the terminology we've thrown at you, you've probably lost focus on the goal. And the goal of electron transport is to generate a proton gradient. So when we talk about incrementally harvesting energy, that energy is going to be stored in the form of the proton gradient. So ultimately, what we're trying to accomplish with these steps is, especially starting with complex 1, the transport of the proton from the matrix to the cytosol of the mitochondria, which involves the oxidation of NADH and the reduction of 1-ubiquinone to ubiquinone in its reduced form, the UQH2, results in the net transport of protons from the matrix side to the cytosolic side of the inner mitochondrial membrane. Once again, what complex 1 is ultimately doing, two things: NADH to ubiquinone and establishing a proton gradient in the inner mitochondrial membrane space. So the cytosolic side, where the protons accumulate, is referred to as the P side, or positive face. Similarly, the matrix side is the N, or negative face, of the inner mitochondrial membrane side space. Some of the energy liberated by the flow of electrons through this complex is used in a coupled process to drive the transport of protons across the membrane, and this happens through a very sophisticated helical piston system which pumps the protons. Now, let me take a shot at illustrating this for you. If we have a membrane where we have a lipid bilayer, and along the top here we have our lipid polar heads and our nonpolar tails pointing inwards, and that's what we're looking at with this simple diagram. If we were to illustrate complex 1, it would look something like this. Where we have a region which lays outside of the membrane, and we have a substantial section which is integrated into the membrane. And what happens here is if you have NADH, which has got our highest energy electrons, it's going to interact with this outside component of complex 1, and it's going to transfer this high energy hydrogen or hydride equivalent plus a proton to FMN, which will then become protonated to FMNH2, which can then subsequently go through a transfer to a pair of iron-sulfur centers. And then these protons can be transferred to ubiquinone, which can then either be recycled or pumped out the other side. So we end up with a hydrogen here and a hydrogen here, two on one side, and then we have a pair of hydrogens in the form of protons on the opposite side. Now, if this takes place, we have two electrons in play. We still have these two electrons. Let's see if I can grab a color. It's not liking it. Okay, we're just going to keep rolling with it. These two electrons can enter another sequence of iron-sulfur centers, which also pump two more protons from one side to the inner mitochondrial membrane space, and then kick out the same two electrons, which can then continue through the system. So what we're really seeing here is the ability per NADH to move 4 protons across the membrane, forming a proton gradient. What complex 2 does is it oxidizes succinate and reduces coenzyme Q. So complex 2 is probably better known by the name succinate dehydrogenase. Oh my goodness, what is going on with this function? There we go. And in your gut you should say, man, I've heard of that. And yes, you have, because it's part of the TCA cycle, and it happens to be the only non-soluble enzyme of the TCA cycle. It's membrane bound, and it was the link between the TCA cycle and electron transport. And what does it do? It converts succinate to fumarate. And as part of that transaction, makes an equivalent of FADH2. So, what does that look like in terms of motion of electrons and generation of ubiquinone? Well, same type of deal. if we have a membrane and we've got our succinate dehydrogenase enzyme, which is integrated into the membrane, succinate interacts with the enzyme being converted to fumarate as part of TCA, and as part of doing this, converts an equivalent of FAD to FADH2. Those high energy electrons for the FADH2 can then go through a sequence of downward steps with iron centers and a heme group, or transfer a pair of hydrogens to ubiquinone. And that's really what we're seeing here, where our sequence, I'm going to oversimplify and say that if we have an iron 2 and an iron 3, we actually need 2 of these because these are single electron transfers. For these high energy electrons, that the ubiquinone will pick up these protons, these high energy electrons, and make a pool of ubiquinone. And this ubiquinone can then be picked up by complex 3. And at this point, you're probably thinking, my goodness, Dr. McGill, what on earth is ubiquinone, and how does it pick up electrons? I'm so glad you asked. So, ubiquinone is a mobile electron carrier. You need some way to move electrons from complexes 1 and 2 to complex 3 in an efficient fashion, which allows you to take a whole lot of NADH and a whole lot of FADH2 and concentrate it in a fashion which can funnel efficiently to a third complex. And ubiquinone is this tool. It's a mobile electron carrier. It has an isoprene tail, which makes it highly hydrophobic, and it diffuses freely in the hydrophobic core of the inner mitochondrial membrane. So as a result, it shuttles electrons from complexes 1 and 2 to complex 3 within the membranous system. So it's a lipid-soluble mobile electron carrier. And what it looks like is this. And this R group is an isoprene tail, which I'm not going to illustrate in detail. Now, what's great about ubiquinone is it has the ability to pick up and release electrons and protons. And what that looks like is reversibly, it can pick up an electron and a proton. And what would happen for that is you get a semiquinone intermediate. So if I illustrate this as UQ, we get a semiquinone intermediate, which involves an oxygen radical, and we get full conjugation of the semiquinone intermediate, and the hydrogen gets picked up by the opposite oxygen in the form of a hydroxyl, and I haven't left myself much room, unfortunately. And this semiquinone can be illustrated with UQH radical. And then you can have a second transfer which satisfies the radical and moves the second hydrogen to the oxygen with the radical position. And retains conjugation. If I reduce this, we can see the whole thing. You can see, oh, and this would be our UQH2. This is how ubiquinone functions. And as you can see, it can grab electrons and protons from complexes 1 and 2 and consolidate them into UQH2, and then it can also release them into complex 3, returning to be UQ, which can then go back to complexes 1 and 2 and pick up more electrons and more protons as a mobile lipid-soluble carrier. So here we are at complex 3. What complex 3 does is it mediates the electron transport from ubiquinone to cytochrome C. And by doing so, also contributes to establishing the proton gradient, which is essential for making ATP, capturing all this energy. Now, it does this through a unique redox pathway known as the Q cycle, which is highly described in the textbook, and I'm not going to go into gruesome detail on that particular cycle. However, generally, it involves 3 different cytochromes and an iron-sulfur protein. In the cytochromes of these and similar complexes, the iron atom at the center of the porphyrin ring cycle between the iron 2 and the iron 3 states, or the ferrous and ferric states. So, like we discussed, at the center of these, when we're discussing transfers of electrons, we have our iron 2 and iron 3. I had an inorganic professor as an undergraduate who routinely liked to state that the most important part of biology was in fact metals. And she wasn't really wrong. The metals really are at the heart of a lot of these electron transfers and our ability to sequentially extract energy from high energy electrons and harness it in a fashion which contribute to this proton gradient, which allows for ATP synthesis. And all of the organic lattice construct which surrounds is about facilitating and regulating and giving us control over process, but with the absence, in the absence of these metals, we couldn't hold on to the electrons the way we do. So let's get into a little bit of detail. Now, what's neat about the Q cycle, even though I'm not going to fully illustrate it, is that it's an unbalanced proton pump, meaning that we're able to actually move more protons across the membrane because of the source of protons coming from ubiquinone than we're bringing from the opposite side via this particular complex 3, that is. So there's 2 different halves to the Q cycle. In the first half, if we have complex 3 protein inserted into a membrane, we'll have this pool of ubiquinone And what happens is that in the first cycle, with no proton coming from the opposite side, we'll get 2 protons into the inner mitochondrial membrane space, as well as electrons moving from the ubiquinone to our cytochrome C. In the second part of this, we get an additional 2 protons coming in and an additional 2 going out. So for 2 in, we're going to get 4 out. Now let me make a little bit of space here. At this point, you're probably thinking, that's really neat. We're pumping protons, we're establishing this gradient, everything is good. What is cytochrome C? Cytochrome C is a mobile electron carrier. And you might be thinking, but ubiquinone is a mobile electron carrier. And you're correct, but ubiquinone is a lipid-soluble electron carrier which moves the electrons from complexes 1 and 2 to complex 3, keeping them in the membranous space. Electrons traversing complex 3 are passed through cytochrome C1 to, which is one of the cytochromes internally, to cytochrome C. And cytochrome C is the only one of the mitochondrial cytochromes that is water soluble. So cytochrome C is water soluble. So ubiquinone was our lipid soluble mobile electron carrier. Cytochrome C is our water soluble mobile electron carrier. It has a globular structure and has a planar heme which lies near the center of the small protein and surrounded predominantly by hydrophobic amino acid residues. The iron in the porphyrin ring internally is coordinated both to histidine nitrogen and to sulfur atoms of a methionine residue. And coordination of the ligands in this manner on both sides of the porphyrin plane preclude the binding of oxygen and other ligands, which is really important because most of the time when you have these structures, you're binding oxygen in soluble proteins. So it's highly adapted to only working as part of electron transport. So cytochrome C, like ubiquinone, is a mobile electron carrier, and it's associated loosely with the inner mitochondrial membrane in the inner membrane space, or the cytosolic side of the inner membrane. And it migrates along the membrane surface in the reduced state, carrying electrons to cytochrome C oxidase, the 4th complex. So, in the same way that ubiquinone took electrons from complexes 1 and 2 to complex 3, cytochrome C coordinates and facilitates the mobility of electrons from complex 3 to complex 4. So finally, complex 4. Complex 4 transfers electrons from cytochrome C, our aqueous soluble mobile electron carrier, to reduced oxygen on the matrix side of the inner mitochondrial membrane. And complex 4 is also called cytochrome C oxidase for this very reason. Because it accepts electrons from cytochrome C and directs them to our 4-electron reduction of molecular oxygen to form water. And what this looks like in a balanced reaction is if you have 4 equivalents of cytochrome C with its iron 2 state plus 4 protons plus molecular oxygen, you can have 4 equivalents of cytochrome C with the iron 3 state plus 2 water. And therefore, cytochrome C and molecular oxygen are the final destination for the electrons derived from the oxygen, from the oxidation of food material. So we start with our carbohydrates, our lipids, our proteins, and they funnel via digestion into their constituent components, and those then make it to glycolysis, and then those get fed into DCA, and that produces a bunch of NADH and FADH2, and then that pumps all the way through electron transport, and by the end of it, the electrons have been converted into water. In concert with this process, cytochrome C oxidase also drives transport of protons across the membrane as well. So just like in complexes 1 and 3, but not in 2, complex 4 will also help establish our proton gradient. So what's this look like? Let me zoom up a little bit here. Without getting into the nitty-gritty of how electrons are moving from component of the protein to component of the protein. If I have a pair of cytochrome Cs, which have bound our complex 4, we can take these two electrons and pass them down through a sequence of copper and cytochromes. And just kind of bounce these around. And then when we get to our final cytochrome A3, which includes the copper, there's a final transfer where our half equivalent of molecular oxygen plus 2 protons gets converted to water by picking up these electrons at this particular step. And while this is happening, 2 protons are pumped because of harvesting the energy through these successive steps. Now, let's try to tie this all together so we can see the bigger picture. Let me do a quick erase, and then we'll call this lecture 1 complete. If we have our lipid bilayer, we've got this wild complex 1, we've got, make a little bit of space here because I'm already running out of room, we've got complex 3, no, don't worry, I didn't skip 2. On the other side we have complex 2, and then we have complex 4. What's happening with these playing together in concert? Well, NADH plus a proton will interact initially with complex 1, giving us NAD+, and as a byproduct we'll get 2 very important things. 1, we will move 4 protons across the membrane to form our gradient. So this is our high concentration side of our gradient. This is our low concentration side of our gradient. That was our first outcome, 4 protons get bumped. In addition to that, we're going to take ubiquinone and transfer our energy to it in the form of high energy electrons. And this ubiquinone is then free as our lipid electron mobile carrier to take them to complex 3. That should be clearly, my goodness, I can't. 1, 2, 3. Now, complex 2 doesn't utilize the NADH. It utilizes succinate to fumarate, forming FADH2. As part of our TCA cycle. And it doesn't pump protons because it doesn't have enough energy. Remember, FADH2 is a lower amount of energy than NADH. So instead of pumping protons with it, we are going to recapture it in the form of ubiquinone, which can then be used by complex 3. Now, the ubiquinone migrates in the membrane to complex 3, which also participates in two very important things. The first is it pumps protons asymmetrically. So 2 protons from the low concentration side to 4 on the high concentration side, and terminally transfers electrons to our cytochrome C, our aqueous soluble mobile electron carrier. And this is now going to be in its oxidized state, and that can then migrate to complex 4, and then back again once it's in its reduced state. And complex 4 is going to terminally give us, from oxygen and 2 protons, water, as our very stable, low energy terminus for electron transfer, and also give us 2 more protons. And this is how the 4 complexes and the 2 mobile electron carriers play together to harvest energy from our highest energy sources, NADH and FADH2, and sequentially transfers the electrons stepwise in a fashion which allows us to harvest the energy in the form of establishing a proton gradient in a very, very efficient manner and squeeze as much of the potential from the high energy electrons as we take them all the way through the electron transport chain as possible. Now, for the next lecture, what we're going to talk about is how does this proton gradient drive the synthesis of ATP. A couple really important points about that. We're going to talk about the interplay of how this was discovered. We're going to tie the entire system back together again from what we just did. And we're also going to talk a little bit about how electrons of cytosolic NADH are fed into electron transport using the malate aspartate shuttle. So that's going to make a good second lecture. I'm going to do that tomorrow and post it for you tomorrow. But in the meantime, I think this is a good place to get started. So enjoy, and I'll be speaking to you about the rest of the chapter shortly.